see my presentation quite clearly right now. You should also hear me. So good morning, good afternoon, good evening, everyone. It's a pleasure to be here with you. And I was invited by Andrzej Miziolek, who is also co-author of this presentation, because he really motivated us and gave us some vision to work on plant analysis and especially to go for cannabis, which is quite interesting topic, and also create a market that is being created across the United States and also in the Czech Republic. So I will be speaking about plant analysis in general by laser-induced breakdown spectroscopy, but I will just focus mainly on the cannabis here. So I'm from the Czech Republic, as you can see, from SATEC. This is a research institute. Also, I'm partly representing Litigo company. You will see some of this, and also there is spectral analyzer from Andrzej, this company that he has. If you have some questions, there is my email, and I will just get into my presentation right now. Okay, I will just briefly go through the outline, just giving some motivation, just what we are now, at what point we are with the performance of LIBS, how LIBS can really contribute to the analysis of plants. Then I will just do some overview and some really quick deep dive into the experiment, what we are doing, how we are doing it, and what are the results. And then I will just conclude our recent findings. As you can see here, there is some publication which is quite recent, and this is some sort of a cookbook for LIBS enthusiasts and everyone who is starting with LIBS and doing some bioimaging that can guide you really step by step to do quite nice elemental imaging. We are doing some upconversion nanoparticles in this case. I will just maybe explain why later. So the motivation right now, especially related to cannabis, feel free to just really extend what I'm just speaking about here, because I'm just really quite new to this field. I'm just more on the LIBS side and providing this service in elemental analysis. So cannabis is really interesting as a plant for bioremediation, phytoremediation, meaning that growing in some soil will just get out these polluted toxic metals and can just be then really used for some other purposes after harvesting. And also what we are speaking here about is just using cannabis in healthcare. And just I really would like to thank here as well to the NIST and NIST representatives that just did this contest on analysis of cannabinoids. It was really a hard time for us to get cannabis samples or hemp samples to the Czech Republic through the customs, but we managed in the end. It was just on the very last minute, so we were able to provide at least some results, but it was really a hard time. Anyway, in the end, we just also were thinking about this and started to grow hemp and cannabis on our own in the lab. So just heads up, we are able to just to do something on our own, and it's just being grown in the lab by our students. So what we are doing here, we are thinking always about in our pipelines. What we are doing is we are trying to think about toxicity and toxicology and how different toxic metals are affecting the growth of different model plants. Before we did really different model plants from radish to mustard to other really corn, such as those really well-established toxicologic plants. And now we're also trying to think more about this cannabis and how actually the increased content of lead and cadmium, for instance, is influencing the growth of cannabis as such. So we just, from the literature research, as you can see also some links here, the hemp is really tolerant to heavy metals. So we will see that in the presentation, and the low heavy metal concentration being present in the soil has no effect on the growth. And we will see that also confirmed by some tests. we are speaking always about toxicology, and we are just complementing the pipeline, the standard toxicity tests that I will be speaking about later, and we are doing mapping, meaning like some really elemental images. This is LIBS is really famous for this, and I will be also sharing some images on my own. And apart of other benefits that you can get from LIBS, the mapping and large-scale analysis is really something crucial in the LIBS community, and we are really proud of this as well. As you can see here, just as time goes by and the systems are getting better, so with that, the number of spectra, the pixels and points being analyzed in one map, in one plant, are just getting higher. So what you can see here, it's just really recent results, and you can see some trend that the spot size is getting lower. Just really the lateral resolution is getting better, reaching 25 microns in a spot, which is really quite nice result, and really getting biologically relevant also for other studies, and getting over 500,000 pixels. And we just submitted a publication where we have more than 1 million pixels in elemental image, and this hopefully is going to be published soon. So as you can see, LIBS can be a really boost in your analysis when speaking about imaging and elemental mapping. So just briefly, we are doing a lot of stuff about analysis of plants, and there are some review publications. You can just go through them. I'm just showing them to just do some showing them to you as some sort of starting point, let's put it this way, to get some really overview of what's happening in LIBS in terms of imaging of plants and also other tissues, as you can see in the review by Andrzej Limbeck and other co-authors. So that's actually just the beginning, and I'm just going to be speaking about what are we going and what are we doing when doing this toxicologic test. And we start simply with some plant germination, just really from the seedling. We are getting to the whole plant, and as you can see on the image, we just after germination, when the seed gets this small root, we just put it in Eppendorf tube, which contains some liquid suspension. It's either pure water or it's some solution of different contents of either cadmium salt or lead salt. So we are really growing those plants in some suspensions or solutions containing toxic metals. And just as you can see here, we are just doing a really wide range of contents, going from 5 to 750 micromole of those toxic metals or toxic salts. So as you can see, as time goes by, we are doing 72 hours toxicity test, which is a standard way, and in 3 days the plant grows quite a bit, and then we just collect all those plants, dry them, really fix them to be on a flat surface, put them on some epoxy resin, and then we can do really nice.
Hello, my name is Sarah Marina.
Yes.
And my name is Carrie.
I'm sorry. Should I go on? Can you hear?
Yes, please.
Yeah, I'm sorry.
My fault.
Yeah. So we are doing this really flat surface for the LIBS analysis, and also we are doing some replicates because, as I said before, we are complementing other analyses. So we are doing the standard phytotoxicity test. We will see that on the next slide, and also we are doing wet chemistry, meaning that we are doing ICP OES analysis, the inductively coupled plasma optical emission spectroscopy, to really get some reference. What is the total content that was absorbed by the plant throughout its growth? So we just do the wet chemistry on some replicates of the plant. Also, we are just analyzing all those suspensions that were left after the growth, and then we are going to do LIBS. And also LIBS is quasi-destructive, so after the analysis the plant is going to be evaporated. So that's the point. We are just doing either one scan if the sample is really flat, then it's being evaporated after one scan, after one map, or we are just repeating this until there is no plant to be seen on the surface. So we are destroying the whole plant with laser ablation. And speaking about toxicity, we are doing some macroscopic endpoints. This is standard thing that is being done for ages. And as you can see here, what is here is normalized length of the whole plant. And you can just also measure it really by a simple way, having a ruler. And you can see 3 different concentrations here, just control and the lower concentration. And as you can see, using this Mann-Whitney U test, this is a non-parametric test comparing 2 different populations because we are doing the repetitions. So you can see that from macroscopic endpoints, from a macroscopic point of view, there is no significant difference between plants being grown in control, pure water, or low concentration of some cadmium salt. So as you can see, low content of cadmium is really doing nothing to the plant, and we then decided to go really for more extreme concentrations of cadmium chloride. And as you can see, only when you get really to high concentrations that are really extreme for the plant and also for some biological relevance. So then after that you get some significant difference, and only for really high concentration the plant feels some toxicity around it and mitigates its growth. So this is really a significant result that needs to be highlighted here. Also, we did some microscopic endpoint. This is just some intermezzo between cannabis analysis. This is white mustard that we also did in parallel to show that standard plants such as mustard, as I said before, corn, radish, and so on, are really getting influenced by the toxicity for even with lower concentrations. As you can see here, there is a huge significance for 20 micromole of cadmium for this mustard, and there is some significance. This is like the level of the significance here, just given by the star, by the asterisk, and the higher number of asterisks meaning the higher significance. So lead content is not that severe to the plant growth, but there is still some change in the growth, and you can see on the plant by visual comparison that there is something happening, but you are not able to see that by naked eye when just having low concentrations in the cannabis. So we are having this standard toxicity test. We are also working on this ICP-OES. I'm not going to show any results. They are just pending right now, those experiments. And I'm just going to show those I'm just going to show some results that we have from LIBS. As you can see, this is the Litigo Firefly, our system that we have introduced recently by our company. And what is worth mentioning is that we are now using different lasers and so on. But in this case, we used 532 second harmonic of Nd:YAG, 20 millijoule per pulse, 20 Hz repetition rate. So it's not that fast as it might be. We are also improving this as well. And also the spacing and the lateral resolution is given by the spot size, which is 100 micron. So each plasma, each pixel that you are going to see in the map is just having a 100 micron size in real on the plant. We are also doing some air purge to just blow away plasma that is being created. We are having different spectrometers, or actually we are just centering this Cherny-Turner spectrometer either on cadmium or lead spectral line, and there is some experimental details, and then we are processing everything in the LITIGO image lab, which is powered by Epina, and you will see some nice pictures from this. And I'm just really going to the end of what I'm going to show you, and this is something that is a representative result coming from the LIBS imaging. And as you can see, you can really image the whole plant. We are just really making this analysis as big as you can see. It's 58,000 pixels. This is quite small map that we are just doing really on. This is as a routine for us right now. And with such a low repetition rate laser, it takes some time. And as you can see, it's almost 50 minutes for this map, but it's quite nothing compared to other analyses such as laser ablation, ICP, MS, OES. So that's quite an advantage for LIBS, that it's quite fast, despite that I'm showing only 20 Hz repetition rate. And as you can see here, the cadmium is just only distributed within the root. It might also, for higher concentrations, it might also get through the root stem to the leaves, but it's not being seen for higher for this low concentration. So we are showing the map only for 5 micromole of cadmium salt, and as you can see, despite that it shows no significant influence of toxicity, it's still accumulating some cadmium within its root, and it might be quite severe when there is some use, such as medical care and so on. So it should be really taken care of, and I'm just showing this as some sort of our application, but I'm surely for medical care there are such much higher standards for safety and so on. And as I've been talking about lead as well, so we are doing also analysis in lead. So you can see another map for the lead image, and as you can see here, we just analyzed the whole microscopic slide size image, and this is 5 micromole of lead salt, and we are having more than 100,000 pixels. This is quite nice map in the end, and as you can see, again, in this case, the lead was mainly in the root, which is obvious because we are just growing those plants only when the root is immersed in the solution. But also in this case, the lead penetrates through the stem. So even for such a small concentration, the lead is already getting dragged out from the root, and it's just getting to other higher parts of the plant. Again, you can see the scale and so on. So you can see that LIBS can give you some really interesting results in terms of imaging. So before I conclude what I was just presenting, I was also mentioning that we are doing not only the toxicity of heavy metals, but we are also doing some toxicity of nanoparticles, and you can see it in our many publications. Also, I just pointed out some of them in the reviews, and this is for apparent reason that nanoparticles are getting in the environment. They are just being applied almost everywhere right now, and just they are secondary getting into the environment, and they are getting everywhere, from plants to also some small organisms. We are also working right now on the imaging of soft tissues, of small fish samples, to just really see lead and cadmium in small fish samples. But this is more difficult also due to some ethical issues that needs to be solved first before the analysis of fish and other animals. So to conclude my talk, I think that LIBS really shows that it's high throughput and can just give a large scale analysis of the whole plant, and you can image using this LIBS technique the whole plant from root to leaf with really high spatial resolution, just below 20 microns. Right now we are on the edge of between 15 to 20 with other systems that we are using for other analyses, such as mouse kidneys and mouse teeth, and also we have quite high throughput with other lasers that we are using. So as I've been showing those maps, LIBS shows that really even for low contents, the cadmium and lead are getting to not just to the root but also to the stem and just proliferates throughout the plant. And this has to be highlighted that and it was just confirmed and it's related to the literature research that industrial hemp is really a robust plant which withstands a lot when it's being grown in toxic environments. So you should be careful about this. And
4 minutes.
yes, thank you very much. I'm just really at the end. So you should be really careful about what's happening around when you're just growing the plant, because you are not able to see that with your naked eye that is being affected by some toxic metals. Unlike mustard or other plants, they are just really getting brown when just being grown in toxic metals and some toxic environment, and you can see it straight away even for low concentrations. So this is just an order of magnitude, or even higher. The hemp is really more robust than other plants. So there has to be some awareness about this. So in the end, I would like to thank my colleagues and co-authors. As I mentioned, Andrzej, he just really gave us a lot of inspiration and ideas what to do with cannabis analysis, and is really pushing us forward in this. And also mainly I would like to acknowledge the work of my colleague Pavlina, Modlitbova, and also my students Karolina and Theresia. They are just doing tremendous work and a lot of analysis, and also my boss Jozef Kaiser, who you might know, most of you. Also, I'm just acknowledging some funding. There are multiple projects that we are working on. If you are interested, it's not just plant analysis. We are just way beyond this. So you can just also take a look on the website, which you can see down on the screen, and there are multiple of different partners in crime. Let's put it this way. And also I would like to thank you to your attention, and I'm open to discussion. Thank you very much.
Well, thank you, Pavel.